All right, so last time we were looking at the basic cardiac cycle, and we kind of talked about how the heart was refilling. It's in diastole, and the majority of the time the heart is just relaxed and blood is returning to the heart. Um, and then, as the blood pours back in, for the majority of it just kind of goes through the atria down into the ventricles. And then when the heartbeat starts is when the atria contract, atrial systole, squish like the last bit of blood into the ventricles. And then after a few moments, you've got the ventricular systole, which is where you get the really, heavy push of blood out to the body. The coordination of all this is done from within the heart. So what we need to talk about now is the intrinsic conduction system of the heart. And this is going to be what we're looking at in the EKG lab later today as well. So let's. So the basic idea here is that the heart has everything it needs within itself to keep beating. If you just went and ripped somebody's heart out of their chest and held it in your hand, it would just keep beating on its own outside of the body, until the tissue dies. It's going to last. If you've ever gutted a fish and you slice a fish open right after you've killed it and the heart still beats, it's really weird. I've never done it in a human, obviously, but in a fish, I can say it definitely just keeps beating in your hand. Part of the reason this works is cardiac muscle is autorhythmic. So let's talk a little bit about cardiac muscle. Autorhythmic means it's going to actually contract in a rhythm on its own without any input from the outside at all. The way this works is it has this like leakage current. So if I do like a graph and I look at, the membrane voltage on this cardiac muscle cell, and remember we've got that's minus 55 threshold. It's like millivolts. Basically what's going to happen is, normally, minus 70, you're kind of at resting. It starts drifting. It just drifts up and up and up and up until it hits threshold. And once it hits threshold, it's just like a regular muscle cell. It's going to have an action potential across the sarcolemma, which is going to trigger contraction. And then you have a resolution of that action potential, but then it just starts drifting up again. And then it hits that threshold and it goes again. So they call this like the pacemaker potential. This idea that the sarcolemma is just going to keep rising up on its own just because hits threshold, you get an action potential and a contraction, and then it resolves, but then it just starts all over again, and it just goes on and on and on and on. The action potential in a cardiac muscle cell is a little elongated compared to what we've been seeing. This tends to last about 200 milliseconds. They think that's so you don't get tetanus. Like you don't obviously remember tetanus. If you kept having firing and exciting a muscle cell again and again, it just had this maximal sustained contraction, which would be a disaster in your heart. You don't want the ventricles to squeeze and never let go. You're not going to pump any blood. So this has a cardiac muscle cell squeeze, but then it has this extended plateau, so you don't start having this kind of summation, and it just then relaxes and gets ready for the next one. So cardiac muscle on its own is autorhythmic. If you just cut a chunk out of your heart and put it in your hand, that little chunk would go whoosh, whoosh, whoosh. And different parts of the heart have different rhythms. In general, they're pretty slow. The natural rhythm of like some ventricle is down in the tens of beats per second. The way that the whole heartbeat is coordinated is there's one special set of cells that go faster and kind of entrain and get all the rest of the heart muscle kind of all coordinated in the same pattern. And that is going to be this intrinsic conduction system of the heart. So let's look at that. Basic idea, let me do red because it's the heart. You've got the heart. You've got the left atrium and the left ventricle, the right atrium and the right ventricle. Up in the wall of the right ventricle, you have the thing that's called the sinoatrial node. It's the part that coordinates the whole heartbeat. Also known as the AV node. This is also called the pacemaker. So this is the little node that is going to coordinate the heartbeat. This goes, about, with no input at all, it goes about 100 beats per second. Typically, obviously, most people's heartbeat is not 100 beats per second unless you're starting to work out or you're pretty unhealthy. What is going to be slowing this down? What controls, it doesn't actually generate the heartbeat, but what can modulate the speed of the heartbeat?
Parasympathetic?
Yeah, exactly, parasympathetic. So the vagus nerve, which is coming from that craniosacral, vagus nerve is one of the cranial nerves, cranial nerve 10. The vagus nerve comes and has parasympathetic innervation and typically slows this down when you're at rest and to whatever your resting heartbeat is. But if you sever the vagus nerve, your heartbeat will rise up to about 100 beats per second based on the kind of just intrinsic rhythm that this little pacemaker node is just going to keep going. Zzz, 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 zzz. Cardiac muscle cells, like smooth muscle cells, are connected by gap junctions. I should probably write that here as well as a I actually have to set the stage here. So first things first, cardiac muscle cells are connected by gap junctions. What is that going to mean functionally?
Co-excitatory?
It means that exactly, they excite each other. If you excite one, they all start exciting their neighbors. So when you have this SA node fire off, I want to erase that. When you have that SA node fire off, you have all the neighboring cells in the atria start all getting excited as well. The excitation doesn't go all the way to the ventricles um, automatically because there is this insulating layer, which we need to talk about. This is part of what we call the fibrous skeleton of the heart. It kind of anchors the valves, and it also electrically insulates the atria from the ventricles. So that's what I'm kind of drawing in blue here. A lot of the meat of the heart is cardiac muscle, but there's also connective tissue in it to help reinforce it. The cardiac muscle and that fibrous stuff together is called the myocardium. So this fibrous skeleton of the heart, we have this electrical isolation between the cardiac muscle cells and the atria and the ventricles. So when the SA node goes off, it's going to co-excite all of the atria. The atria, that's going to be that atrial systole, where the atria contract and swoosh blood down into the ventricles. But we don't want that excitation to immediately go to the ventricles, because otherwise, they'd be pushing back. You can't actually push the blood from the atria to the ventricles if the ventricles are pushing back up at you at the same time. But then obviously we do want the ventricles to contract for the main ventricular systole. So there's going to be another node. It's called the AV node. It's kind of right here, kind of up near this boundary between the atria and ventricles. So the AV node, atrioventricular node.
Can you repeat what makes up the myocardium? It's this.
It's the myocardium is just going to be so it's the cardiac muscle and the reinforcing connective tissue in there.
Okay.
If you've ever, if anybody's ever eaten, if you can eat, beef heart is actually super yummy. It's more meat. It's muscle. It's mostly muscle.
The first node should be SA node, not AV node.
Oh. You are so right. Thank you very much for catching that. So SA node is the pacemaker. AV node is the one that is going to excite the muscle cells, the cardiac muscle in the ventricles. Obviously, we need to have the excitation of the SA node make it to the AV node. So there is going to be this what's called the intranodal pathway. Not intramodal. Come on, don't autocorrect me. Pathway. And it has a delay. Obviously, like I said, you do not want the ventricles going right away, because then they would push back and you couldn't actually move the blood from the atria to the ventricles. So it's usually like an 0.1 to 0.2 second delay. So this is going to be something we're going to see in our EKG in a few moments. Or not. So, once the AV node goes off, it is going to excite all the ventricular muscle. if you just let the gap junctions do their job, eventually the AV node would excite all the ventricles, but it would be kind of a slower kind of thing, like, right? You'd have to like, and then these excite these, and then these excite these, and then these excite these. It would be kind of slow. So there's actually a set of they're actually specialized cardiac muscle cells, but that are optimized just to transmit the electrical signals quickly. There's ones that go right down in between the ventricles called the bundle of His. Then that bundle of His splits into the right and left bundle branches that are going to the two sides. And then those just continue into these things called Purkinje fibers. Like one of the heart conditions that you can have is called bundle branch block. If you have bundle branch block, it means that the signal is not transmitting well down the bundle branches. The ventricles will still contract, but rather than doing a nice, clean, tight, instead it's like, right? Because it means it's going to take longer for that excitation and contraction to spread across the entire ventricle instead of having it being distributed really quickly through these conducting fibers. So this is the basic, in fact, I can probably, give me 2 seconds here. If I go into remote instruction, where'd it go? Where'd it go? Here we go. Like, so here I can, just to try to make it prettier for you for a second, I'm going to share. So here, this is showing it in a prettier way than I could just draw on my screen. So again, this whole thing is, again, at first the heart is just relaxed and refilling, blood is coming in. Then when everything is going to start, you've got this SA node, this pacemaker node is going to send out, it's going to depolarize. When it depolarizes, the atria will contract. And then there is that short delay. This intranodal pathway, which is going to take, again, about 1 to 2/10 of a second. And after that 1 to 2/10 of a second, the AV node fires off. We got the bundle of His, the bundle branches, all the Purkinje fibers, and that's going to cause the ventricles to contract. So that is kind of the intrinsic conduction system of the heart. I think this thing does a little, it's like, ooh, look, we contract the atria. Then after 1 to 2/10 of a second, the AV node goes off. That then is going to be transmitted down the bundle of His and the bundle branches, the Purkinje fibers. The other thing that's kind of cool here is these little things. These are called the papillary muscles, and these are called the chordae tendineae. There's actually this little reinforcing thing, the little heartstrings. This actually pulls down on the flaps of the valves during ventricular contraction to reinforce them, to make sure that they don't blow back, because there's a lot of pressure. So these little papillary muscles and chordae tendineae actually help pull and reinforce the flaps to make sure the flaps of the valve don't blow back. You know, what do we call it if blood does gurgle back up into the, gets back up even though it's not supposed to? These valves should only allow blood to go from the atria to the ventricles, but sometimes the blood can leak and gurgle back up. What do we call that?
A prolapse?
But more, if you have, you can hear, so there is that as well, but you can hear the noise of the, like when people have like a heart murmur. A heart murmur is, you can either have blood leaking through the AV valves or leaking through the semilunar valves to get a heart murmur. And one of the, actually this ties into our whole learning to take the blood pressure, but one of the kind of non-intuitive things, think about having a louder murmur versus a quieter murmur. And you might think that like loud is worse, but in general, it's not true. Loud can also mean it's barely whole at all. So it's very turbulent, but it makes like, you can hear it. If the leak is worse, it's actually going to be quieter because it's not going to be as turbulent. So a quiet heart murmur doesn't mean it's not as bad. It actually can be worse because for the same way that when the brachial artery was not as compressed, you didn't hear as much noise. You know, if the leak is worse and the blood has even more smooth flow through the really bad leak, you're not going to hear as much noise. So electrocardiogram. So this is just the electrical signals that you can measure on your body that are generated by the depolarization of the cardiac muscle. So as the cardiac muscle depolarizes and contracts, there are these big voltage changes. And if you just stick electrodes on, in our lab, we just put them on your two wrists and you can measure pretty big voltages that are very easy to see. There are lots of different ways to measure. Again, this is ECG if you're like in America. EKG is electrocardiogram, like more Germanic. It's the same thing. It just means an electrical heart picture, basically. It's the muscle depolarizations as measured on your skin. When you look at it happening, it basically, there's lots of different ways you can do it. There's much more fancy ones, 12 leads, where you put little leads all over and get much more subtle information. But at the core of it, it looks like this. You see a first bump, you see a boom, 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 and then another bump. And it goes, wait a while, boom, 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 boom. Kind of keeps going like that. The first bump, this is called the T wave. This is called the QRS complex. Not the T wave, P wave. And this is the T wave here. So the P wave is from the beginning of the whole thing. What is happening at the very beginning of that cardiac, when the heartbeat starts? Remember the SA node, the pacemaker node, is starting the whole thing. What is the big electrical thing that happens in response to the SA node firing? What does it excite? Exactly. So this is going to be atrial depolarization. So atrial depolarization, as that electrical excitation spreads across the atria, it shows up measured on your skin as this bump that we call the P wave. For this QRS complex, this is going to be when the next big voltage change happens. What's the next big voltage change that we're going to see?
Ventricles depolarized.
Exactly. This is ventricular depolarization. So, and if we think about what we've been talking about, if you look, I'm going to erase this here. If I look what we call like the PR interval, how long should that take? What should that time be from that P to the R showing up?
0.1 to 0.2 seconds.
Exactly. And we're actually, that's part of our lab today is normally you'd measure it on yourself, but instead we'll look at it on Tina. And you'll get to make sure that that looks right. So then the last part of this thing is the T wave. So so the T wave is actually representing ventricular repolarization. So this is after the ventricles have depolarized, contracted, and then when they go back to resting, you know, it goes up because you're not if you had an electrode stuck in the heart, it would go up during depolarization, down for repolarization. But because you're actually measuring the electrical signal on your skin, you know, basically you're a big bag of salt water with this voltage shifting heart in the middle of it. This is what it looks like measured on your skin. So don't worry that it's going up, even though repolarization means coming back down. This is just the way it actually shows up when you get to all of the electrical calculations of what does it mean to have this heart beating in a big bag of salt water. So this is the electrocardiogram. Like when we take our break after lecture before lab, you should probably, you know, review this and make sure this makes sense. P wave, QRS complex, T wave, because that's going to be at the core of a lot of what we're doing in lab is we're going to stick electrodes on the subject and see what we measure. And it looks like this. What do I not have in this picture that you might wonder about? We have atrial depolarization. We have ventricular depolarization, ventricular repolarization.
Atrial repolarized.
Yeah, exactly. Atrial repolarization. So obviously it happens, but it gets lost. The QRS complex, the ventricular depolarization is really big and massive. So the repolarization of the atria gets lost in the QRS complex. So this does repolarize, but you don't see it. So that is why it's missing. I can give you people who are really good at interpreting electrocardiograms can tell you so much stuff. Ultimately, the EKG is only electrical information. Electrical. So it doesn't really tell you if the ventricles are contracting, or, you know, if I see, all I can, you know, it could be you've got some horrible scar tissue or whatever, and you actually have the muscle depolarizing, but you're actually not really squeezing, you know, because of whatever. So it's not actually telling you ventricles contract. It's saying the ventricles are depolarizing. But assuming everything's working right, it can tell you that it's contracting. People who are really good at interpreting these things, especially with the multi-lead, can actually tell quite a bit about heart function. That's a whole class. It's something I don't know much about even. I've looked at some of the stuff. There are a few things that do show up that we actually have seen in our lab. Tina does not show, luckily, but I can show you kinds of things that are kind of easy to see that go wrong. One thing is this. All right, maybe if I show it. So what's like going on here? Yeah, normally if you have a P wave, what comes next?
The complex?
Say it again.
The complex?
Yeah, the QRS. P wave, and then 1 to 2/10 of a second later, you should see a QRS complex. Sometimes you see a P wave and then nothing happens. And then the P wave happens and then it looks normal. Again, usually it has, yeah, unless this person is in a lot of, is in real danger. Most of the time it's doing that, but every once in a while you get a P wave and nothing happens, and then it kind of goes back. So what's going wrong if the P wave fires off, but then you don't get the QRS complex?
The signal. The depolarization isn't reaching the ventricles.
Exactly. So this is going to be a problem with the internodal pathway. You know, so sometimes the internodal pathway can be intermittent. Most of the time it works, but every once in a while the SA node fires, but it doesn't actually go to the AV node and start the second part of the heartbeat. Like I said, this is common enough that I see it every once in a while in lab. You know, as long as it's just intermittent, it just means every once in a while the heart skips a beat. Other things that we see that are not that uncommon. So what is going on here? Why does this look wrong? Remember, it normally looks like this. How is this looking different than this?
It's taking longer.
Yeah, it's taking longer. It's all spread out. So this would be typical kind of heartbeat if you have like a bundle branch block. The ventricles do eventually depolarize, but it takes a lot longer for that depolarization to spread across all of the ventricle. And again, this happens. again, this is not something that is going to kill you. Again, we've seen students in the class who exhibit this. What happens if the coordination isn't happening? We get what's called fibrillation. Let's talk about fibrillation briefly. So this is when you have kind of uncoordinated contraction, of cardiac muscle. Like I said, cardiac muscle on its own has autorhythmic properties. It goes, part of the reason why it all works fine is the pacemaker node. That SA node is basically reining everything in and driving everything and coordinating it all so it all works together. But if something goes wrong and you lose that coordination, you just get kind of quivering of it's if you have, sometimes they say like, uncoordinated quivering, like, so if we have ventricular fibrillation, uncoordinated quivering of the ventricles. That's really freaking bad. The ventricles are supposed to squeeze and generate the pressure that pushes the blood out into your body. If they're just sitting there going, the blood is just splooshing around and you're going to die pretty quick because you're not generating the core blood pressure to keep your blood flowing. So if someone's in ventricular fibrillation, you want to try to get that resolved really quick. What is the basic way to try to deal with ventricular fibrillation?
Install a pacemaker.
Ultimately, you're going to need a pacemaker if there's problems, but in the moment, this person's going to die pretty soon if you don't get the heart beating again.
But you shock someone with a defibrillator?
Yeah, you use the AED, that defibrillator, those paddles, like, all right, ready? Yeah. Like, what are they doing there? It's basically like if your computer freezes. And I don't know why it's frozen. It's like this morning, it's like my audio didn't work. And I don't freaking know why it doesn't work. All I know is if I turn it off and turn it on and let it reboot, it's working again. So putting those paddles on someone and putting that big shock across them is basically kind of like doing the power off, power back on, and hoping it reboots right. And hopefully the heartbeat is now coordinated again and working properly. So ventricular fibrillation, if you're in ventricular fibrillation, that's really kind of an acute problem because you're not pushing the blood out into the body. Another kind of fibrillation that's not uncommon atrial fibrillation is not such a horrible concern in the moment, right? Because we talked about, as the blood returns to the heart, most of it, 85% of the blood just pours through the atria into the ventricles. And so by the time the heartbeat starts, 85% of the blood is already in the ventricles. So if the atria aren't good at coordinating, at least pushing blood into the ventricles, it isn't really going to be tragic. You're still going to be fine. But the problem that can happen in atrial fibrillation is the blood ends up kind of pooling and it slooshes around in here and it doesn't keep flowing. And when that happens, it's more likely that you're going to end up having a thrombus, a blood clot forming as the blood just kind of sits around and doesn't keep moving. And And then if that eventually does get pushed into circulation, it's going to go and block your circulation somewhere and give you a stroke or something. So people who have atrial fibrillation, they tend to put them on blood thinners to make it less likely that they're going to have some blood clot that forms that's going to cause problems. If you have really bad atrial fibrillation, they also do kind of this electrical thing to try to bring it back into normal beat. But again, ventricular fibrillation, if you don't fix it, you're going to die in the next couple of minutes. Atrial fibrillation, this is actually not uncommon for people to have. So what's the time? So that is all this electrical stuff. Electrical stuff about the heart. And again, which we're going to be looking at in our class or in our lab later. But now what we're going to do is we're going to go back and start talking about kind of the fluid dynamics of blood flow. So now back to blood pressure, etc. So a lot of what we're going to talk about for the rest of today is ways your body helps keep the blood pressure regulated. If you remember, blood pressure is cardiac output times resistance. Again, cardiac output, this was just our measure of flow. So when we think about ways your body can control blood pressure, it can do it by controlling cardiac output or by controlling resistance. Resistance, we've already talked about. What's the primary way your body can adjust resistance to blood flow?
Changing the diameter of the vessel.
Exactly. Change the diameter of the vessel. You constrict it a little more, the resistance goes up. You dilate it more, the resistance goes down. So that is something we've already kind of looked at. Check. Cardiac output. This is something we need to spend some time now talking about in more detail. So we are going to look at cardiac output in more detail and what are the things that affect it and what are the things the body can do to change it. So again, we'll still be coming back ultimately to this equation because we're looking at ways to regulate blood pressure. But to regulate blood pressure, we have to regulate blood flow. So let's look at that. So cardiac output. Does anyone remember what is the definition of cardiac output?
The volume of blood pumped by each ventricle per minute.
Yeah, so this is going to be liters per minute pumped by either ventricle. And again, this is our measure of flow. So when we think about cardiac output, it is going to be stroke volume times heart rate. So let's talk about these now. Stroke volume. This is liters pumped per beat. So how much blood is swooshed out of a ventricle in one beat? Heart rate is just what you think about. This is like beats per minute. So stroke volume. So cardiac output is liters per minute pumped by either ventricle. Stroke volume is going to be just how much the liters pumped in a single beat by a ventricle, by either ventricle, I should say. So let us, cardiac output is going to be stroke volume times heart rate. So heart rate is, so right, if you go, this is going to be liters per beat times beat per minute. The beats go away, and that's going to give us our liters per minute. Right, so this all kind of makes sense if you're doing the basic math here. Right, the beats cancel. So heart rate, this one is easy. You can speed up or slow down the heart rate with the parasympathetic or sympathetic nervous system and change the frequency of the sinoatrial, the pacemaker node, and put the heartbeat up or down. Stroke volume. This one we need to look at in more detail. There's a lot of math in looking at how blood pressure. So we talked, blood pressure is cardiac output times resistance. Cardiac output is stroke volume times heart rate. Stroke volume is now going to be further broken down into things that the body can deal with. Let's look at that. It's going to be, let me write this better. Heck. I'm going to write it in typing so it is easier. So it's going to be something that we call end diastolic volume. Minus end systolic volume. Yes. So let us see what this means here. So our stroke volume is going to be end diastolic volume. So remember, diastole is that refilling. The heart is relaxed and refilling. So this is how much blood is in the ventricle at the end of diastole while the heart is being refilling. So this is kind of the volume in the ventricle as the heartbeat starts. As ventricular systole starts. So the heart has been refilling during diastole. At the end of diastole, right before the ventricles are going to squeeze, this is whatever the volume is there that's there to squeeze out. And systolic volume, so systole is the squeezing. At the end of the squeezing, it hasn't squeezed out every last drop that was there. There's some blood left. And so this is, how much is left at the end of the contraction. How much left in ventricle at the end of when contraction is over. Right, so the amount of blood that you squeezed out, the volume in the beat is going to be how much blood was there to start with minus how much blood was still left over after we were done. Right? So in fact, I'll give you actual numbers. Doobie-doobie-doo. Where is my, here we go. So let me give you some typical numbers here. I'm going to erase this stuff. So a typical number for end diastolic volume, 135 mils. So that's how much blood is in the ventricle as the heartbeat starts, or as a ventricular, I should say, as a ventricular systole starts. Usually there's about 65 mils left over at the end of the heartbeat. So what does that mean my stroke volume is going to be typically?
$70.
Yes, 70 mils. So now 70 mils is a typical stroke volume. We can go back up to this equation here. So I'm going to erase this stuff. Go back to this. So I can say, all right, cardiac output, typical. A typical stroke volume is going to be 70 mils per beat. A typical heart rate. Most of you all as kind of young, fit people, it's probably a little less than this, but the number people usually use for a typical heart rate for a person is about 70 beats per minute. So what does that mean in terms of a typical cardiac output for somebody? How many liters per minute are coming out of either ventricle?
$490.
Getting close. So 7 times 7 is going to be 49. Adding 2 zeros on it for each of those.
4.9.
4.9, exactly. So it's around, if we round 4.9, it's basically 5 liters per minute. So a typical cardiac output. Right now, most of you just sitting there in front of your computer has your heart, we can go again. You've got your heart is here, squishing blood out to your body through the aorta, putting blood out to your lungs through the pulmonary trunk. Both of these are currently spewing out, somewhere around 5 liters per minute going out into each of the circuits. Remember, we said that cardiac output has to be equal in the pulmonary and systemic circuit. So these are both spewing out 5 liters per minute. You can increase that pretty dramatically. So this is at rest. So we call this the cardiac reserve. Cardiac reserve, can increase 4 to 5 times, up to 7 times, in a conditioned athlete. So that's pretty dramatic. That means just for a normal person, you can have but 5 times 5, 25 liters per minute coming out of either ventricle. Conditioned athlete, 35 liters per minute. A liter, I mean, oh, do I have a liter? I don't have a liter on me. You know what a liter is, though? I mean, a liter is about like a quart. In fact, I do have a liter. Like, imagine like 35 amounts of this coming out of your heart in 1 minute. It's crazy. It's got this amazing ability to really increase the flow to supply all of your tissues. So what we need to talk about is, how can you do that? How can you increase cardiac output by 4 to 5? So this is cardiac reserve, is increasing cardiac output. So it can increase cardiac output. So we can go back to our picture here. There's only a few things we can work with. So let's look at what we can work with. So what are going to be ways to increase cardiac output?
Increase the heart rate.
Totally. Increase the heart rate. So if we increase the heart rate, then you're going to increase cardiac output. So that's going to be an important way to increase cardiac output, is increasing the heart rate. And again, if we take ourselves, just to kind of keep us in context here, remember, by increasing the heart rate and increasing the flow, you're helping increase the blood pressure. I can just tell you a personal experience. One time I was cleaning out my compost, and there was a huge cloud of these spores that came up. And I was just breathed in and totally enveloped in this huge cloud of spores. And I had some kind of allergic reaction to it. And I ended up turning red. I think I had just major vasodilation everywhere. So my resistance to flow went way down because all my blood vessels dilated as part of this reaction. And what I noticed was my heart rate just went through the roof. It was actually really scary. All of a sudden, my heart was like boom, 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 boom. And I actually had a little automatic blood pressure cuff at home, and I checked myself, and my blood pressure was in a safe zone. But I realized what was happening was that because the resistance had went down really far, that my body was compensating by having to increase the cardiac output to keep my blood pressure from plummeting. And one of the ways it was doing that was by like doubling my heart rate. And all of a sudden my heart rate was like, I don't remember, I think it was somewhere like 140 or something. Like for a few hours, it stayed really high before kind of the, reaction went away and everything started going back to normal. But just in a kind of real world example, if the resistance went down because of vasodilation across my body to this allergic reaction, one of the ways my body kept me from dying was increasing the flow to keep the blood pressure up, and a way to increase the flow and to increase that cardiac output was just to increase the heart rate. So that is one way to do it, is heart rate. The other one, obviously, we can adjust the stroke volume. Again, this is going to be the end diastolic volume. minus the end systolic volume. So here we have two things to play with. What are ways we could increase stroke volume with these two? Again, this was end diastolic. This was end systolic volume. So how can I increase stroke volume using either of these?
Increase the end diastolic.
Yeah, exactly. We could either increase end diastolic or we can decrease end systolic. And so both of those are things your body does do. Let's talk about this one first. This is going to be easier to explain. So you can increase this, which means that there's less left in the ventricle at the end, which means there was a higher stroke volume. We've squeezed more out. To squeeze more blood out of the ventricle, it means you got to squeeze harder. We talked about skeletal muscle was this kind of all or none kind of thing. Cardiac muscle is different. You can actually have graded contraction strength with cardiac muscle. For instance, the presence of epinephrine, of adrenaline, it binds to receptors on the cardiac muscle cell, and the same stimulation of that muscle cell can result in a stronger contraction than you would have had if the epinephrine was not there. So you can increase the contractile strength by having, for instance, adrenaline present. Increased contraction strength means you squeeze more out, which means that you, this is not increase, you decrease. You have decreased end systolic volume, like for instance, by stronger contraction. You know, for instance, with EG, like for example, like if you have epinephrine around, epinephrine will actually create a stronger contraction of the cardiac muscle cell. So the same contraction squeezes more blood out of the ventricle, which means you're increasing stroke volume. You know, the epinephrine is also going to be working over here, right? The epinephrine is also going to be increasing your heart rate, which is going to increase cardiac output. And in reality, if we go back to the blood pressure thing, epinephrine is also going to constrict your blood vessels, increase resistance. So epinephrine is doing lots of different things to bring your blood pressure up. And when you jam someone with an EpiPen, you know, you're doing that basically because they're anaphylactic shock, their blood pressure's dropping down to nothing. It's doing lots of different things to bring their blood pressure back up. It's constricting their vessels, which brings resistance up to bring blood pressure up. Epinephrine is going to increase the contractile strength of the cardiac muscle, which will decrease end systolic volume, increase systolic volume, which helps bring cardiac output, which brings blood pressure up. It increases heart rate, which is going to bring cardiac output and therefore blood pressure up. So epinephrine is doing lots of things, but in this case right here, we're just looking at it affecting end systolic volume. This one is another thing, end diastolic volume. How much blood is in the ventricle when the heartbeat starts? I need to do a little aside here. So the cardiovascular system, it says closed circuit. I'm going to just kind of make it a little bit of a cartoon. You've got let's just look at the systemic side. You've got the arterial side. You've got the veins kind of returning the blood to the heart. Most of the time, you have way more blood kind of in the veins, kind of limping along, making it back. Like the heart is generating pressure to keep the blood flowing really nice and quickly through the arterial. The veins, on the other hand, we're going to see later, there's little valves. It's slowly moving its way back. So normally the majority of your blood flow is in the veins. So more blood flow in the veins compared to the arteries, but you can shift that a bit if you really need to increase the end diastolic volume. So you can increase venous return. You can push more of the venous blood back up onto the other side by constricting the smooth muscle in the veins. So if you constrict the smooth muscle here, that means there's less overall volume for the blood to be kind of hanging out there, which means more of it is getting pushed back up into the heart. And that is another way. If you increase venous return by pushing more of the blood that's in the vein side, it means that you're going to end up having more blood returning to the heart and increasing your end diastolic volume. So at the beginning of the heartbeat here, you'll have a higher end diastolic volume. You know, this is kind of like due to increased venous return. While I have this other picture here, I should bring up one more point. Because you have this closed system like this, all these things we're talking about that affect blood pressure, the resistance, the diameter of the vessels, and the speed of the heartbeat, and this and that, those can all affect blood pressure in the short term, bring it up or down in the short term. But the main long-term control of your resting blood pressure is actually ultimately the volume of blood in this system. So main long-term is, and I'm going to put it like in caps, blood volume. And I'm going to increase the font, and I'm going to like make it like boom, 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 boom. I wish I could animate this so it just keep going, but I can't. But I can like circle it in a highlighted color. So you want to keep this in mind. If you think about it, if there's not enough blood in this closed system, it's going to be kind of more flaccid and it's not going to be as much pressure. If you have too much blood in there, it's going to be pushing against the walls and you'll have too much pressure. So there's kind of an ideal volume of blood given the amount of vessels that you've got. So you can do all these things in the short term to keep the blood pressure up or keep the blood pressure down by constricting vessels or speeding or slowing up the heart. But for the kind of long-term control of your resting blood pressure, it's the volume of the blood that's the most important thing. You know, this is going to be, what is the organ that ultimately is adjusting your blood volume?
Kidneys.
The kidneys. So actually not today, probably on Monday when we continue talking about blood pressure control, we're going to see a big part of blood pressure control has nothing to do with the blood vessels of the heart. It has to do with talking to the kidneys and telling them to change the amount of water you're keeping in or letting go in your body. Again, like important drugs for blood pressure control are diuretics, things that make you pee more, right? Because if you have high blood pressure, one of the ways to try to bring it down is peeing more, get more water out of your body, bring the blood volume down. That's going to help bring the blood pressure down. So we will be talking much more about this, but for right now, let's get back to where we were. So stroke volume, we can control stroke volume by doing end diastolic volume or end systolic volume. So this is talking about blood pressure meds. Let's introduce, but before we're done, we're gonna talk about 3 main kinds of blood pressure meds that are prescribed. There's, again, the diuretics, things that make you pee more. There are the ACE inhibitors, which we're gonna talk about next class. And then there's beta blockers, which we can talk about right now. Let's make it fatter. So beta blockers, it's actually a fancy way of saying beta adrenergic antagonists. So what is a beta adrenergic antagonist? What does that mean? What is adrenergic? What's an antagonist?
It blocks epinephrine.
Exactly. This is going to block epinephrine. And how is blocking epinephrine going to help keep your blood pressure down? Remember, blood pressure is cardiac output times resistance. What does epinephrine do to your heart rate?
It increases it.
It increases it. So if you're blocking epinephrine, you're gonna then be able to not have this increase of the heart rate, keep the heart rate down. We also talked about how epinephrine can actually affect stroke volume by increasing the contractility of the cardiac muscle, decreasing end systolic volume, and therefore increasing stroke volume. So when we block epinephrine, we're also keeping the stroke volume down. So both of these things are keeping the cardiac output down, and keeping cardiac output down is keeping the blood pressure down. You know, we're also going to later on, epinephrine is also going to be constricting blood vessels and keeping, so by stopping the constriction of blood vessels, you're also keeping the resistance down. So that should all make sense to you, like blocking epinephrine, keeping your heart rate down, keeping stroke volume down by reducing the contractility of the cardiac muscle and therefore decreasing end systolic volume, which keeps the stroke volume down. Both of those keep cardiac output down. Cardiac output down keeps the blood pressure down. Now, over the weekend, when you're starting to think about all of this, kind of write it down on a piece of paper that, this kind of equation is kind of at the top of the heap. And then cardiac output is this. And then stroke volume is going to be, end diastolic minus end systolic. So there's all these, this goes to this, but this is affected by it. So ultimately, though, you're seeing how adjusting this is adjusting your blood pressure. So it might look all confusing and like a bunch of crazy, luckily they're all really simple equations. They're just like pluses and minuses and multiplies and divides. But you do want to kind of arrange them all and make sure you kind of understand how they all work together to control your blood pressure, because that's going to be an important part of this part of the course is, and we're going to get into a couple of other things that control blood pressure next class. But for right now, it's 9:30.